Let me move on, and the problem has been temporarily resolved. I am very happy here today. With us is Professor Kristi Anseth. Professor Anseth is a distinguished professor of chemical and biological engineering from the University of Colorado at Boulder, where she is also associate professor of surgery, as well as professors by courtesy of chemistry and biochemistry, as well as molecular, cellular, and development biology. She has been named by the AICHG as among the 100 chemical engineer of the modern era, and so she is one of the best in our profession. From the titles, you may have already figured out that Professor Anseth's work is at the intersection of science, engineering, and medicine. She has accomplished at a very young age and selected being one of the few who have been selected as to the all three branches of national academies in the USA, including the National Academy of Engineering, National Academy of Science, as well as the Institute of Medicine. Professor Anseth's interdisciplinary research combines physics, chemistry, biology, as well as mathematical modeling to design biomaterials for clinically relevant applications, including drug delivery and tissue engineering. As a matter of fact, I have always selected works from her group as reading assignments for one of the chemical engineering elective class. And that's because the approaches she's using is always very systematic, logical, and illustrates really how an engineer should think about biological problems and how an engineer can come up with elegant solutions to these problems. So Professor Anseth has received numerous awards, including the Alan T. Waterman Award from the U.S. National Science Foundation, the Alan P. Coburn Award from AICHG. Her teaching also has been recognized by Curtis McCraw Award from the American Society for Engineering Education. In grad school, I was a friend of one of her former students, and behind her back even, he spoke really highly of Professor Anseth, especially noting her dedication to research and her devotion to her students. So I think that should be one of the best recognitions for a good teacher and a good mentor. So with that, I will pass the times to Professor Kristi Anseth.
Well, thank you for the very kind introduction, and had a delightful time visiting the campus this past day, and I'm looking forward to the next couple of days and learning more about all the exciting things going on here. What I decided to do today was to give a little bit of an overview, a general audience type of talk that introduces this area that we've been very interested in, and that's how do you regenerate tissues, how do you engineer tissues, and get the body to heal itself when processes go awry. So I entitled my talk the Body Shop, because I think oftentimes we can all think about different replacement body parts we might like to have. And what I'm going to do is begin with just a little bit of introduction and motivation, kind of set a stage, our context of how we think about this problem, what we think are some challenges from the field, and walk you through a little bit of a historical perspective of how things have evolved and where they're going, advances that have been made, and what might come in the future. So let's take a step back, though, and let's go back several thousand years ago. And when you began to think about some of the ways that biomaterials have played a role in medicine, so many thousands of years ago, people didn't understand what caused disease. And many times when there are injuries, the main thing was to try to salvage life. So that led to, infections weren't known, and so they would bleed people. A severe injury, they might amputate a limb. So the idea was salvaging life, but even 3,000 years ago, they found on this mummy, if you can see up close, this person had their toe amputated, and a wooden toe was used as a replacement body part. The large toe, the big toe, is very important in our balance. So even many thousands of years ago, they're beginning to think about using ways to restore tissues in the body. Now let's fast forward several thousands of years now. And we have many different types of wonderful medical devices that not just make people's lives better and last longer, improve quality of life. And we have things like replacement joints when they become arthritic, so patients can walk and move those joints without pain. Or if you have heart valve disease, different types of synthetic replacement valves that allow those people to live. And so thousands of examples, pacemakers, different types of imaging systems. And so what I'd like to talk about, though, is while these have been made very wonderful strides and are widely impacting people's lives, if you look at them, many of the materials and the structures of these are very different from the natural tissues which they're replacing. And they're all designed to be a permanent replacement, but that's still one of the challenges of the field, is that these devices wear out and they have a limited lifetime. So the idea is, where might things go in the future of medicine, and how might engineering principles help with this? So how can we begin to think about no longer synthetic replacement body parts, but strategies and ways to intervene to get healing to occur? And so the idea isn't necessarily so new. One part of our body that's very, very important are all the cells that reside throughout our tissues. And so cells become a very important part of the therapy in trying to regenerate tissues. And there are many different cell delivery and cell therapies in clinical medicine. Blood transfusions have been around for decades, saving people's lives when there's large amounts of blood loss and we can't regenerate our blood cells fast enough, so we deliver them. Bone marrow transplants are used to treat many types of cancers. Islet transplantations, so islets produce insulin and when you're diabetic, your cells no longer can properly regulate glucose in your body. And if you have really severe diabetes that can't be adequately regulated by insulin injections, the Edmonton protocol now exists where they'll take from a donor islets and transplant that into the patient with the goal of trying to restore better control of their body's glucose. I'll talk a little bit about this one, autologous chondrocyte implantation. That's taking your own cartilage cells from somewhere in your body and trying to regrow or repair cartilage so you don't have to have those synthetic joint replacements. And in fact, if you do a search of clinical trials, and these numbers are just for the US, so you can imagine what they're like worldwide, there are tens of thousands of clinical trials that are based upon using cells to get the body to heal itself. Stem cells delivered to patients' hearts after a heart attack, trying to regenerate the muscle. Different types of neural cells delivered to patients' brains, trying to eliminate debilitating effects of disease such as Parkinson's. But despite all these different trials and advances, these cell-based therapies, I'd like to propose that they're growing, and I think they're going to have increasing impact in medicine for treating disease and injuries. But many times, when you just inject the cells all by themselves, only a few numbers engraft. So that means that they survive in the tissue and they have some type of functional output. So many times, the number of cells that engraft might be only a couple of %, very low. So the idea is, could we engineer a second part to this? Because many, many cells in our body reside in tissues and organs, and they're surrounded by a matrix. And they get really important signals from that matrix. And so you might think of it as a material environment, a niche. So how might we exploit engineering principles and biomaterials to help with this? And that's really a lot of what the tissue engineering community has been about in the last 25 years or so, where the basic idea or concept is to take cells and to grow them on different types of material environments in 3 dimensions. And one example here is if you take fibroblasts in your skin and them on woven fibers of degradable sutures, the fibroblasts will start to proliferate and grow. This is an actual picture. And quite amazingly, they'll begin to regenerate a tissue structure that resembles aspects of skin. And the degradable sutures dissolve away. And in fact, this was the very first clinical product, a tissue-engineered skin that's used to treat patients that have diabetic ulcers or if they have severe burns over a large fraction of their body. So the idea in the field is, so how do we sort of integrate knowledge of the cells and their properties and functions and the signals that are needed to guide them with engineering principles and scaffolding type of environments that we can put these cells in an appropriate context and deliver those signals in the appropriate way in a way that addresses problems and collaborations that go over to clinical medicine so that we might design actual products. Now, the part that we work in is right in this area. And one of the problems of the earlier stages of the field is that when you looked at the repertoire of different types of biomaterials that I could use to transplant for human medicine is that many biomaterials were never designed as a biomaterial to become in contact with the human body. Instead, they became a biomaterial when someone went through an iterative process of implanting that material and seeing what happened. Now, this next slide is from a former postdoc mentor who kind of summarized some of these biomaterials that we have approved. So things like the first artificial heart made out of ladies' girdles materials. Here's the polymer ether urethane. A vascular graft used for bypass surgery is made from Dacron, which you're more likely to make socks out of. So even to this day, one of the big problems in cardiovascular tissues is that many of the biomaterials cause blood clots to form. It still is a problem in the field. Those implants that I was talking about for joint replacements, a lot of times those surfaces wear, and that's a really big problem. Sometimes they've used things like Teflon as a low-friction surface that many of us maybe know as a cooking surface. But one of the things is that, unfortunately, Teflon doesn't stand up to wear very well. And so that's caused problems with different types of implants. So the idea is, how do we go about trying to rationally design different types of bioscaffolds that can serve as carriers for cells and in many times the drugs that are necessary to signal to those cells so that you can regenerate a functional tissue or improve survival and engraftment of cells when you deliver them. And so many of these requirements are things that are such as biocompatibility in the context of which it's being used. Most of the time, we'd like these materials to degrade so that you're left with just the natural functional equivalent of that patient's tissue. A lot of times, the engineering side, you need to worry about the physical and mechanical signals and transport properties to these cells, especially as you have very complex three-dimensional structures. And I say provide instructions to the cells as needed. Those instructions oftentimes come in the form of different types of growth factors, proteins, small molecules, and so the chemistry of with which the cells need to receive. Now, the type of bioscaffold that I'd like to focus most of my lecture on today is one that we've had a lot of interest in, and that's hydrogel type of systems. So here's just a macroscopic picture of a hydrogel. If you could see the molecular level details of a hydrogel, what you'd be able to see is long macromolecules that normally would just dissolve in water. But those long chains have interactions between them, and those interactions render it insoluble, a solid. So the hydrogel imbibes large amounts of water because of the chemistry of those long molecules, but the interactions between the chain make it a gel that has many properties that are very desirable for culturing cells. So you can embed cells in these types of hydrogel environments, and the hydrogel properties render many soft tissue-like properties that are found in natural tissue environments. And the large amount of water inside of these allows cells to get signals that you might be delivering to them, as well as signals that the cells are secreting or depositing when they're making those tissues. So oftentimes it's all about figuring out and engineering presentation of those signals, how many are critical for the process, what time scale do they need to be delivered over, as you're trying to coordinate or facilitate this complex series of events. Now, one way that we sometimes think about these hydrogels is, on the one hand, we think about them as a completely permissive environment. So what do I mean by that? It allows me to culture cells in 3 dimensions in any shape that I would like. Cells might get some physical signals from the surrounding, maybe mechanics of the environment. There can be some mechanical signal. But in general, I'm just providing these cells with an environment with which they can survive, proliferate, secrete matrix molecules that might be important for that tissue. And I'm making the cells do all the work. And I'm just providing a physical structure. On the other hand, you can think of these hydrogels as something that becomes very promoting. That I want to give the cells a certain signal, to tell them to move in a certain direction, to tell them to differentiate to a certain cell type, or maybe to stimulate them to form tissues faster. And so now here I worry about the chemistry of the environment and the specific interactions that I'm giving to the cells. For that I need more information, and I need to know which signals to give in what context. So what I'm going to do is I want to walk you through a little bit of an evolution where we started working on this side of the equation, where we're working with permissive type of hydrogels. And I'll walk through an example with regenerating cartilage, but then tell you how we needed to become more sophisticated as we looked at and evolved with different applications and directions. So when we began our work in this field, one of the areas that we were really interested in was regenerating cartilage tissue. So cartilage is a tissue that's on all the ends of our bones at articulating joints. And they provide cushioning and a low friction surface, and it allows us to move those joints without pain. But unfortunately, cartilage has a fairly limited capacity to heal itself when it's injured, has a low blood supply, a low nerve supply, and when it gets injured, you alter the biomechanics of the joint, and when the biomechanics of that articulating surface are altered, you can get wear. And so it might take several decades if you injured yourself downhill skiing in your 20s. Maybe by the time you're 50 or 60, the cartilage surface has worn down to the bone, and when that occurs, now it becomes very painful to move the joint. So this affects many different people, and the recommended treatment is to remove this whole joint, saw off the ends of this joint, and put it in a synthetic replacement joint, the picture I showed you at the beginning. But the problem is that if you're very young, those joints can't last your entire life. So you have to look at multiple replacements. So what I'd like to share with you is one story. We had a collaboration with an orthopedic surgeon who was treating a 41-year-old woman who had a very severe cartilage defect, and she really did not want to have a total knee replacement, but yet she had this debilitating knee pain. So I'm going to show you a picture of what a cartilage defect looks like in an open surgery. So if you don't want to see this, just look down, and I'll tell you when it's done. Okay. So here is the cartilage defect. That's healthy cartilage. That's the patient's kneecap. Okay, it's away for those of you that are. So state of the art for trying to get cartilage to regrow about 15 years ago or so was to take a biopsy of the patient's own cartilage tissue, because she has lots of healthy cartilage cells. To isolate those cells and expand them, so those cells are the chondrocytes, and then to re-inject them back into the patient's knee with the hopes that those cells would regenerate or regrow the cartilage tissue. They're healthy cells. I just need to put them at the site where the defect is. So these are what the biopsies look like. So they remove the cartilage plugs, and then they're sent away by the surgeon. And then I'm going to show you one more picture of what this looks like. So here it comes. So this tissue called the periosteum is removed from the bone, and it's sutured along here, and then the cells are injected underneath here. Okay, so the picture's away. So the problem is that procedure of taking the patient's own cells and redelivering them works okay if the defect is really small. But when the defect is really large, like what we saw, it's very difficult to regenerate cartilage tissue throughout that. And the unfortunate problem is that most people go see their orthopedic surgeons when they have a huge cartilage defect or large loss across a large degeneration of their cartilage tissue because they're in pain. So the idea here was that maybe resurfacing of cartilage joints could work better if we designed a material carrier for them. So here's cartilage tissue. So let's look at this picture. If I slice up a piece of cartilage and I stain it, what you'll see, so these are the cells, the chondrocytes, and they reside in these little spherical lacunae in the tissue. And these cells are responsible for making and maintaining the cartilage matrix, and that consists primarily of collagen. And it's interesting, it's one of the few tissues where it's type 2 collagen. And these are the mechanical struts that give the tissue a lot of its rigidity and strength. Proteoglycans are the second main component, and these are branched macromolecules that have sulfated disaccharides along the edges of this, and that leads for the tissue to imbibe large amounts of water. So cartilage has lots of water, so this allows the tissue to be compressed and recover from many different cycles of use. And so the blue here is staining for the collagen part, and the red here is staining for the proteoglycan part. There's lots of water here, but to us, this seemed like a good strategy to design a hydrogel to deliver those chondrocytes. So our idea was we would design an injectable system with cell delivery that we would solidify inside of the defect, and so then once we had a hydrogel laden with chondrocytes, that hydrogel would give the three-dimensional context to those cells, maintain them in their lacunae, and the cells already know how to make these matrix molecules. So hopefully over time, we could regenerate healthy articular cartilage, and the gel would degrade away. So for those of you interested in the chemistry, I'll show the structure here. Those of you not, I'll try to go quickly. But the design of the hydrogel, we use this middle molecule. It's polyethylene glycol, PEG. One of the most widely used synthetic polymers in human medicine. It's very hydrophilic. So that was one of the reasons we chose this molecule. And then we just grafted off really short units of lactides. That's what degradable sutures are made of. So this would break down in the body at physiological pH. And then we put these end groups on. And so these are the ends that we're going to be able to trigger a reaction, and they'll link up. So this will start out water-soluble. I suspend the cells in this water-soluble solution. We happen to use light to catalyze this reaction, and it'll form a solid gel. And when it does, you can entrap chondrocytes inside of that gel, and they take on the spherical morphology, much like in the native tissue. And one of the reasons they take on that spherical morphology is that this chemistry, the cells don't recognize it all. They're not interacting with it. Okay, so now we've made this hydrogel. The types of things we want to engineer or think about are the properties of this hydrogel. So you can think of it as a mesh in 3 dimensions, and depending upon the size of that mesh, I change the properties of the gel. So I can change things like the water content of my gel. So this is water over here. The elastic modulus of the gel, how stiff it is. And those all depend upon that mesh size. And so the tricky part about hydrogels is that all these properties are all interdependent on this one property of the mesh size. So even diffusion coefficients, so if the cells are going to make this tissue, how those molecules can diffuse through the hydrogel will also depend upon this mesh size as well. And so it's this trade-off. So water content goes down as I make it tighter mesh, but thing that I'd really like to have if I'm going to put this in somebody's load-bearing joint. And so we kind of were looking at, could we optimize this? And this yellow region is about what articular cartilage is at small deformations. So that's what we did. We entrapped these chondrocytes in gels with different types of physical properties. And now we sectioned them, we cultured them for a period of a few weeks. And then we looked at the cells. They survive. Now here we're staining for that one proteoglycan molecule. And what you'll see here is that I go to a tighter mesh. Here are the cells. And that molecule the cells are secreting can't diffuse far away. And it's all just in the paracellular region. But over here, if I open that up, that molecule can distribute very facilely. And as engineers, we also like to model this. We can construct different types of molecules based upon diffusion coefficients and how fast the cells secrete that molecule. And so this would be representative of something like this. And then we change our diffusion coefficient, and we can kind of do thought experiments to figure out what experimental space we should design our hydrogels. But the tricky part really was looking at collagen, which is one of the really important molecules for mechanical reinforcement of the tissue. And when we looked at collagen, even in this system, that's the blue stain here, and these are the cells, and this is just the gel, and there's no collagen anywhere in this gel. The problem is that collagen is a nanofiber type of system, and it's made and synthesized through a complex series of reactions of the cells secreting different procollagen molecules have to assemble into these large fiber diameters. And so it's really hard to make a hydrogel system that allows that process to occur, and instead the gel really has to break down and lose mass to allow this process. And so here I won't go through as much details, but I had a student who did a lot of work in characterizing the hydrolysis of those linkers that we put in the gel and how those gels lose mass. He did a lot of optimization, and so you can kind of see here the gel degrades much too slowly. And then you have to worry here because if the gel degrades too quickly and dissolves, then the cells haven't made enough tissue and you're left with cartilage tissue that has lots and lots of defects. But you can sort of optimize this, and we learned a lot about regions within this mass loss curve where a lot of the collagen gets elaborated. And so over the years, it took about 5 to 6 years, a couple of PhD students, we were able to translate this into a large animal study, and this was a collaboration with a group at Johns Hopkins and a long-time collaboration with Jennifer Alicieff, who's a bioengineer there, where we would create defects, and here's a goat's knee joint, and we could deliver our cells to this goat's knee joint and come back 4 weeks later and see large volumes of regenerated articular cartilage. And in fact, if you look at and section that cartilage, this is what it looks like in the native cartilage, and here's our regenerated cartilage. So even architecturally, the structure looks a lot similar to the regenerated tissue and the native tissue. So this takes us through many years of work, and it was licensed and gone into human clinical trials with a company, Cartilex. And about 20 or 30 patients were treated in a European clinical trial with this procedure and approach. They were followed for 12 months and monitored with MRI, and a large volume of tissue was regenerated in the defect. And then many measures of pain and joint mobility were done and sustained over this 12-month period. This was very exciting. It was to get something into human medicine. But the tricky part here is that ultimately the story failed, and Cartilex was unable to raise funding to expand their clinical trial. Part of it was our timing, that it was just with the economy, and so that we're unable to raise the funds for that, even though the technology worked. But the other thing is that we had to rethink and go back a little bit because the technology is more expensive than a total knee replacement or total hip replacement. And there's lots of talk in the US and other countries of trying to get under control costs, medical costs. So in the US, at least our healthcare budget, there's been an extreme inflation of how much of our GDP we spend on healthcare. And so introducing technologies, it's a technology that works. You can regenerate cartilage in people's joints. But you have to think about it in the context of other products and other things and how it would be reimbursed. And so that made us go back and think in more recent years, how can we change our vision here? Because what makes that expensive is that it's a personalized system for everyone. That it's 2 surgeries, I have to get your own biopsy, expand those cells, you come back for a second surgery to have that put in place. It's really difficult to know if it will work for somebody, the same for somebody who's 60 versus 20, designing a product for that. There could be differences in gender. And so the idea was, well, maybe we should be thinking about different types of material systems and healing that perhaps we would either deliver something that might be a progenitor cell population that would be on bank or be available, or perhaps we could just deliver a material itself, and maybe a material with some drugs as well that could recruit resident cells. And so that would simplify some of the ideas of the procedure. But now we had to think now about promoting gels, because no longer could we just permissively deliver healthy cells from the patient that already have the ability to make the tissue. Instead, I have to either recruit cells or use a population of cells that I might have to direct what type of tissue to form. And so while our permissive cells were really great for chondrocytes, many cells that you might want to recruit from the body are in niches that require lots of signals from the hydrogel system. So let me see if I can do this. How do I escape? There we go. Oh, here. Okay. So the problem is that many of the cells that require signals from the tissue environment, if I put them in permissive gels, here's what happens. Maybe. Okay, so let's watch these. So these cells, they're trying to attach to the gel. They're trying to move around towards one another, becoming very frustrated because they don't recognize any of this chemistry. They're trying to deposit things around here, but nothing sticks to it very well. And so they're eventually going to undergo a programmed cell death called anoikis because of lack of these matrix interactions. So that makes us then rethink what types of tissue systems and gels should we be considering. And so we use the native tissue matrix, the native extracellular matrix, to kind of guide us. So the native extracellular matrix, what kind of signals do cells get from that? Well, they bind to this matrix through adhesive proteins. And sometimes that's very important for survival or for maintaining their stemness if they're in a stem cell niche. Cells also make proteases or enzymes to degrade these matrices. So they decide when they're going to remodel it and deposit new tissue, rather than like the hydrolytic ones that I just pre-engineered to degrade at a certain rate. And it's also a reservoir for many different types of growth factors that the cells, when they degrade the matrix or if there's an injury, that they can bind and release on demand. So while our permissive hydrogels gave the water content and mechanical cues, from another sense, these are kind of some of the minimal properties we were thinking about for the native ECM. Now, you might think, well, why not just assemble cells that you like inside of all the natural matrix molecules found within the tissue niche? Collagen, elastin, other types of components. So that can be a very good approach. The problem is it's very difficult to get those proteins, to purify them, and to reassemble them in a way that makes them functional like the original tissue. At least it's difficult for us in our hands. So we tried to simplify it. And instead, what we did was we used peptides, so smaller sequences in those proteins, to try to capture critical aspects of the protein functionality. So for example, in different types of adhesion proteins, we know exactly where the cell's integrins bind to that. So RGD is found in fibronectin, adhesive protein. Laminin, we know exactly where nerve cells interact with that through IKVAV. We also know cells make many of those different proteases, and this happens to be one that's found in collagen, and the MMPs 1, 2, and 3 will cleave this right here. So we know it can design different types of sequences for different cells for them to degrade. We can also screen through different types of peptide sequences that will bind specific growth factors with affinity. So this one happens to bind TGF-beta and different BMPs with affinity. So that was our idea, and so now we had to think about, well, how are we going to put these peptides inside of these hydrogels? We could synthesize them using just a solid phase peptide synthesizer. And so the different part here was we put the amino acid cysteine into our peptide sequence. And the reason we did that was we wanted this thiol group as our reactive component. And then we still were going to use our PEG to give us a lot of the nice hydrogel properties, but we just changed the end group of this PEG. And the reason that we did that is that this particular end group reacts very efficiently with this particular thiol, the cysteine thiol. If you mix them together, though, what's interesting is they won't react. It's only in the presence of light, and we catalyze it with light through a reactive intermediate. So it's a really interesting reaction chemistry, and you can react these two components together, mix them in different ratios, and click together and form different types of functional hydrogels. So the idea is that I could use my PEG part, its molecular weight, its number of arms to control that mesh size, and I can use my peptide and the sequence that I make for the cells I'm interested in to control the biological signals. I can click those together in different ratios and look for hits in cell properties of interest. Do I want cells to proliferate fast? Do I want them to migrate? Is it a stem cell and I'd like it to differentiate? So I can screen through these fairly rapidly to find a formulation that's suitable for my cell type of interest. And so now I'm just going to show you one particular formulation where we use this RGD, this adhesive ligand, and we use this sequence here, which is degraded pretty rapidly by these bone marrow stem cells that we've been working with. And I'm going to go out of the slideshow again one more time. Here we go. To show you what happens. Maybe? Okay. So I'll try to put these side by side. So on the right here, this is one. It's those cells. Remember those cells that I showed you at the beginning where they really wanted to move around in the PEG gel and they couldn't and they're going to die? So here is a PEG gel, and I just put those peptide parts in there, and the cells are attaching and locally degrading and migrating through that gel. So something that much more closely resembles native tissue. So localized degradation. Now over here on the left, if I misspell that sequence and I put an alanine instead of tryptophan, and these cells have a harder time degrading that, what happens is these cells can survive, they attach, but they can't go anywhere very well because they can't degrade it fast enough. So you can make material niches for different cells and their ability to move through this. Okay, so let's go back to the PowerPoint. Oh yeah, down here. Okay, so if we go back here, so now why is this interesting? So we were interested in thinking, could we get cells to locally infiltrate this gel? I happened to have a medical fellow in my group at the time who was an otolaryngologist. So these are doctors that repair large craniofacial defects in your body. And it's really, really important in trying to repair those defects because your skull protects your brain. So having fast bone regeneration is really important. And so we wanted to test some of these materials where this is a CT scan of a rat skull. So we're looking through the rat skull. And we create what's called a critical size bone defect, 9 millimeters in diameter. And when you have large amounts of bone loss, the bone can't heal itself. So that's the kind of defect he was interested in repairing. And here we put in our hydrogel that was the one that the cells couldn't migrate through very well in that picture. And here's one that they could. They could migrate through this very well. And what was striking to me about this was the large amount of bone healing that occurred in this system, where this is just a material. There's no growth factor, there's no drug, there's no cells that you've delivered. You just intervened here by placing a temporary provisional matrix that cells could infiltrate. And if you look at this, so you can see this, so quite dramatic. In this gel, so you get some bone growth right around the edge. Here's the defect. And then in the gel itself that just allows cells to infiltrate, you see large volumes of bone regenerated. So here's some of the analysis over an 8-week time period. So while this is a very interesting result, though, what I will say is that it takes 8 weeks, and it wasn't perfect healing, right? So you can see some defects here still. So while we've done a good job, now you have to start asking your questions, why doesn't it completely heal? Do I need some type of promoting factor that when the cells infiltrate, they get a signal that maybe tells them to make bone faster, makes it osteoinductive? Or maybe I need some type of chemotactic factor to get more cells to infiltrate, or different types of cells. So with respect to the growth factor approach, I'll just tell you a little story, is that at the time we were doing some of this, recombinant bone morphogenic proteins were one of the most successful areas of recent interest and success in tissue regeneration. So this is a protein, it's a growth factor, and it's known to be very osteoinductive to promote bone regeneration. And so a new product had come out called Infuse, and it was being used to try to help with patients that had problems with their vertebrae, their spine, and they needed to have them fused. And so normally you'd have to take a bone graft from your hip in one surgery, and then they'd use that inside of the cage to get bone to regrow. Sometimes even just removing from the hip causes a lot of pain to the patient. So if they could use a protein, then maybe that could help and facilitate faster and better fusion without having to have that second surgery. One side note, though, is that for one patient, I haven't counted all the people in this room, but if I took all the BMP from your bone marrow, probably wouldn't quite be enough to treat one patient. So it's a really large dose. And the reason for the large dose is that BMPs, it's difficult to deliver and to localize and has more limited solubility. And so they have to give really high doses for this to happen. And one of the problems that happened a few years ago was that this was sort of a product the FDA approved for sort of fusing vertebrae in the lumbar region, your lower back and that area. And they were also starting to use it in the cervical region near your neck to try to fuse those for patients that had problems. But then this led to lots of adverse effects. So what happened was that children that were treated with this had huge inflammatory interactions. And it's in the cervical region, and it cut off their airways and their ability to breathe. So I had to give them steroids, and that didn't quite control it, and they had to go and remove these devices. So the problem was some of the side effects. When you deliver this super physiological dose, you get side effects from the growth factor that leaves. And so now it's no longer approved for this use, and it's only in lower back regions, but it points to the need for improved delivery systems. So the ideas and things about trying to figure out how to deliver molecules from hydrogels. So what we were sort of thinking about at the time was, one, you could just entrap the molecules in the hydrogels. But the problem is, lots of times when you want cells to be able to infiltrate and you have lots of water, that most molecules come out pretty rapidly out of hydrogels. It kind of depends upon the molecular weight of the molecule you want to use. So then another really common way is to make tiny little microparticles out of those degradable sutures and to load them up with different types of growth factors. And so they can be embedded in hydrogels, and those degradable sutures sort of release the protein over longer periods of time. But that chemistry of microparticles, sometimes you can run into problems. Does the activity of my protein stay inside of those microspheres? So we were just thinking of how could I entrap molecules in my hydrogels and try to slow down this release. And so we were thinking like a chemical engineer of a reaction diffusion controlled release. And so what do I mean by that is that you start to put those peptides in that bind growth factors with affinity. So, and again, we were trying to mimic some of the extracellular matrix and how growth factors are normally stored in that. So now when this growth factor is trying to diffuse away, it's going to bump into affinity ligands and it's going to slow its release down. So that's what we were sort of thinking of, and it was pretty amenable to the chemistry we were working with at the time. And so we were using these PEG peptide gels, and you can think of this as this is the unmodified protein. I haven't done anything. And here's doing my Thiol-Ene reaction, forming the gel around it to entrap it. We wanted to make sure we weren't doing any damage here, and we weren't. But that other system that we used for the chondrocytes did lots of damage to the protein. So you have to be careful, depending on your protein of interest. And then we could control the release of proteins by putting in these affinity binding ligands. And as we increase the ligands, we could slow down the release rate quite a bit. So I won't go into lots of details here, but I'll just show you. So we did a sort of a side-by-side comparison. So the infused products takes these BMPs and all of it, and they just absorb it to a collagen sponge. They implant that. And this is the dose that they use, scaled down to the size of a rat. And so when we look at the infused product, you can see it works pretty well. And this is only 6 weeks, so there's lots of healing across here. And here's what's happened when we use our gels that we're promoting to cell infiltration along with the BMP, that could be affinity bound. And for us, what happened was we saw at this dose huge amount of bone growth. These rats looked like they had a football helmet of bone grown on top of their head. And we think, we don't know, but that we probably are localizing the BMPs longer at this site, and it's just causing lots of bone formation that's unwanted. And in fact, we knocked it down a level, an order of magnitude lower. And this is how the infused product does. So it does okay, but that's why they have to go up to these doses. Whereas our system here, even at an order of magnitude lower dose, we still got a large amount of bone healing. Again, this is 6 weeks, so it's faster than you. So it's not perfect. But this shows you one of the challenges of the field is that this is just one growth factor that we're trying to deliver to promote healing. And it's still really tricky to figure out the right time scale and the right doses. And so where we're sort of going, should I stop here or can I, is it okay? Okay, all right. So I just have a couple of more slides. But so where we're trying to think about going is how can we figure out new types of tools that will allow us to better screen through the factors of what dose should I give, what concentrations, what time scales should I release, and also what happens if you need to deliver 2 factors, or let alone 3. So our ideas were, so how can we take permissive types of hydrogels, load them up with different signals that might be needed, release them, remove them, maybe give a different signal, and do this in a high throughput way so I could learn about some of this and then from my in vitro results then test it in vivo. And so I'm going to give this in the context of one last example is it's like what happens if I want to use a more complex progenitor cell type or where I might have to actually deliver multiple cues to this molecule. And so we were collaborating with somebody who was treating patients with Parkinson's disease. And he had done clinical trials where he'd figured out a way to make dopaminergic neurons from precursor cells. So when you have Parkinson's disease, you lose the dopaminergic population within your brain. And so those are the cells that make dopamine, which is responsible for having coordinated muscle movements. The problem is it's really difficult to take a drug to restore that dopamine because it's difficult for many drugs to cross the blood-brain barrier, and this dopamine is made in a certain region within your brain. So he'd figured out where to deliver these cells, how to make the dopaminergic neurons. But the problem is that, again, 1 week post-transplantation, at best, 10% of the cells survive. And that long term, so patients that they had treated and then they looked at longer term after the patients had died and done analysis, that they found very little evidence of any of the cells transplanted surviving. So the idea is, how can we figure out and improve this delivery process and figure out what types of cells are necessary to promote this dopaminergic nerve function? Is it that I need to give it a survival cue? Is it that these neurons need to extend processes in certain directions to integrate with the surrounding tissue so that they can function and store long term? So just very quickly, what we did as a tool for this was we took our same hydrogel design, but now rather than having a cell-degradable crosslinker or a hydrolytic degradable crosslinker, we put in a crosslinker that would degrade with light. And we made these into little hydrogel microspheres that we were going to load up with different types of growth factors or molecules that could inhibit or promote axon extension of neurons. And we could put multiple types of factors inside of our hydrogel. And you can consider these like little nanoliter delivery systems. Now, the thing about these nanoliter delivery systems is that the growth factor is just embedded in this, and it stays there until I expose to light. And then it dissolves the microspheres and releases that on demand. So the idea is I can do different types of thought experiments while I'm watching the neurons in vitro and deliver these different cues and see how they influence the neurons. Does it help them survive? Does it help them extend processes? And let's see, one more movie maybe. So here's the idea. Here's these microspheres. And when I click play, I've just exposed them to a laser, a focal laser beam that's about this big. And the gels begin to degrade, they increase in size, and then they just release in a few seconds their contents. So that's the idea. So we loaded these up. We're culturing little neurospheres of these dopaminergic neurons, and we're beginning to release factors in this periphery by just rastering this laser beam across here. And then you can watch with time. If you see this very well, the processes begin to extend, so I have to give more and more of a dose to figure this out. But by day 14, we can begin to calculate the dose and amount to get robust process extension. And now we can also begin to think about experiments. These are processes in every direction. Maybe I'd like to guide it here and inhibit on this side. And that's what some of the value of these neurosphere structures are. And the last image that I want to leave you with is that the nice thing about hydrogels is when you do these types of experiments, that you can real-time image cells and their function. And so here, what you're watching is calcium signaling between neurons as I'm delivering different types of signals on the outside. And I can see and analyze their coordinated signaling and the types of signaling patterns. And that's what we think is going to be really helpful, is to know whether these neurons get integrated into different surrounding tissues. So with that, so I just wanted to leave you with a few thoughts. I think there's lots of opportunities in trying to use engineering principles, material design, collaboration with people from biology and medicine to try to design systems of tissue engineering, regenerative medicine importance. Our body is a superbly engineered biological system. We have 100 trillion cells. So how can we develop better products and ways to improve quality of life that doesn't just necessarily rely on synthetic replacement parts, but ways to intervene and to tweak systems that have gone awry? And I think that we're seeing lots of growth in cell-based therapies. And the tissue engineering approach has already led to lots of advances. We already have tissue engineered skin. We can get bones to heal faster that won't. We can regenerate cartilage. And there's been lots of advances in tissue engineering blood vessels for bypass grafts. But as we start to think about more complex tissues, translating them to medicine, these are lots of ambitious goals and lots of challenges. So what happens when we have to culture multiple cell types or deliver multiple types of factors? These are challenges that we can solve, but it's going to require lots of collaboration. So many times, identifying the cell source can become difficult, and especially with clinically relevant numbers. The cartilage, that wasn't the problem. Dopaminergic neurons, it definitely is. Trying to solve diabetes, it's a huge problem. Trying to regenerate cardiac tissue after a heart attack is a problem with finding cells. So oftentimes that means using different progenitor cells and culturing them in environments that's important to promote their differentiation, I think can be very helpful. We also maybe need to define what are functional properties of the tissue engineered equivalents. Maybe we're a long, long way from being able to tissue engineer an entire pancreas. Maybe we could regenerate a little functional subunit of an islet that just replaces the ability of glucose responsiveness of those cells, that it can make insulin in response to local glucose levels. Expanding repertoire of biomaterials that can be used in medicine, combined with figuring out how to deliver cues at the right time in the right place. I didn't touch on this too much, but all the legal and regulatory issues as well. One of the biggest problems that we have is what's going to be reimbursed and covered by the healthcare system, and we need to be thinking about that when we're designing our products. So with that, I'd just like to acknowledge great group of students, postdocs, visiting scientists I've had a chance to mentor over the years. So Boulder is a landlocked state in the middle of the US, so I've really appreciated the setting here in Hong Kong. But we do have the beautiful Rocky Mountains. Our university is right at the foothills of the Rocky Mountains. And so everything that I told you about today is a lot of the work and effort of these students. We've also had lots of really nice collaborations throughout the years, and I need to also acknowledge my funding. But with that, I'd be very happy to answer questions.
The floor is now open for questions.
Okay, I'll try to break the ice. Hopefully some other people will ask questions too. So a couple of things struck me thinking about what you've been doing. And you mentioned directing neuron outgrowth. So have you been putting nerve growth factor in or something like that to direct them over in the direction you want them to go?
Yeah, so our original work focused on nerve growth factor, BDNF, GDNF, but now we're also trying to look at things that are maybe opposing gradients. So sometimes ones that will inhibit as well as promote, and gradients of those as well. So we're kind of at the early stages of some of that. But right now we find that when we just singly deliver one of those BDNF, GDNF, or NGF, we get pretty uniform process extension. So we can increase the extension.
But you can't really.
But we can't in one direction yet, yeah.
And do you do anything, well, okay, so I liked your imaging of the nerve cells talking to each other because I like to think about that in C. elegans, right?
Yeah.
And so have you done anything in model organisms like C. elegans or collaborated with people that were to look at how the nerve cells are talking to each other in culture or in the embryo?
No, that's a great idea. So we haven't done anything with C. elegans. What we have done some is, so we've done a lot of our isolations from the mouse or rat mid-forebrain. And so we've done some organotypic cultures where we've sliced it and then done cultures short-term in vitro and looked at that as well. And so we were interested when you deliver what happens to signaling.
So back when I was in Hawaii before I moved to Macau, I did embryonic cell culture in C. elegans, looking mainly at intestinal lineage development with endoreduplication and things like that. But in a total embryonic cell culture, you get neuron outgrowth and cells connecting, and you don't know what they're doing because you don't have any labels on them, right? I think that's a very fertile system for looking at some of these things, as long as you have the right connector signals to be able to watch. And calcium pulsing is clearly cool.
Yeah, that's a great suggestion. Thank you.
I do have that system able to be set up here. I just don't have it.
Yeah, very nice talk. So I have a question. So before you use the hydrogen to deliver the chondrocyte to the cartilage, so what is the older way people use? Just indirectly inject the chondrocyte to the...
Yeah, the original technology is just delivering the chondrocytes. And some of the problem with that is without the three-dimensional context, the chondrocytes just settle and attach to the adjacent cartilage tissue, and that changes their phenotype some too when they're just attached, and they make more of a scar-like cartilage and they don't fill the volume.
All right. So what is the, I didn't quite get the idea of the using the photoactivable.
Yeah.
So is it for more like precise tempo control? Or
yeah, so the idea is using it as an in vitro screen where you can load up a hydrogel with multiple factors of interest, and then you can begin to do thought experiments where I can watch cells in real time and deliver a particular signal and watch how the cells respond to that signal. Or I can deliver two signals and watch what synergistic response it has to those two signals. So the idea is with one simple formulation, I can screen through many factors rapidly and do real-time screening of my cells. And then once I figure out conditions that seem most appropriate, then I can design a system for the in vivo translation, which would deliver those molecules on the right time frames. Otherwise, you're kind of left with, I make a microsphere formulation that delivers at a certain rate, and then I test it, and if it's the wrong rate, I have to reformulate my whole microsphere, put it back in and test that one. If that's the wrong, maybe it's too slow, then I reformulate again to try to get one that's right in between.
So can you like use the photoactivable technique to control deliver for like more than one signal molecules?
Yes, so we kind of color code them so we know which ones are loaded with which molecule. And then you come in with a laser and you basically just deliver the depots that you want that maybe are the closest to your cells. And maybe you pick one factor delivered first and then a second factor.
So how many you can do now simultaneously?
So, so we've loaded up about 5 different factors. Right now, the limitation is that we have to use a laser to degrade them, because the laser, we spatially control which ones degrade, because if we just expose to floodlight, they'd all degrade. Yeah. So we need to make them maybe of different labile linkers if we want to have different wavelength control. Yeah.
Thank you.
Okay. I have two questions here. Thank you very much for the presentation. First one is on the heat replacements. Because of Hong Kong, people live very long. Female, we are war number 1. Male, we are war number 2. So we have a lot of these issues. And I talked to the people in Hong Kong use orthopedics. Right now, they're still doing a very traditional one. Cement with screws that do a bunch of finite element modeling, figure out how many screws, how big is the critical size they can do. They put PMMA and so on and so forth. But I understand the reimbursement that you mentioned. But Hong Kong has also another issue is breast cancers. And breast cancers, after you take away the local tissues, you need to do cosmetic hydro recovery. But that seems to me is where hydrogel would be affordable. Because of, so I'm just wondering first whether apply that technology, which is too expensive for knee replacement, whether been some efforts taken to look into the breast cancer, the region recoveries. That's my first question. And the second question is more on the delivery, which is make the very smart hydrogel systems for different delivery, which I think is a good idea. But how many of those are going to be say, approved in FDA in the near future, particularly when it.
Well, so for example, the peptide functionalized gels I talked about. So right now, that one is, there's a small startup that has phase 1 trials to take that through for approval. But again, it's like PEG-based and peptide-based. So chances are that it should be a pretty compatible system. But what you worry about is if your peptide doesn't completely degrade and a small amount stays attached to the peptide, will you generate an immune response to PEG? Because it just takes a few amino acids to cause that. So that's one thing they have to check really carefully. And then so more advanced things like the photolabile system is probably one you wouldn't necessarily use in vivo. Maybe there might be some niche applications in ocular delivery or things like that where you could have light. But I think the value is that maybe that helps us figure out different proteins that we could deliver because we're still kind of trying to do a better job of understanding how to deliver a single protein and control it. But you could imagine that if I had multiple affinity factors that I could control 2 or 3 at different rates by controlling. Otherwise, like in a single hydrogel system, if I give you 2 proteins of different molecular weight, it's kind of hard to engineer a single system to deliver those independently. So that's what we're kind of thinking with some of the affinity delivery.
I have a general question, actually, 2 general questions. One is about your thoughts on how to take the results from in vitro and then interpret them for in vivo or clinical applications. Because a lot of times, we engineers like to use a very simple systems and then do some screening and then modeling. But then how, in your experience and in your perspectives, like what do you think, whether those results are really valuable or how can we use those results? And then the second question is about actually the issues that you haven't touched on is the regulatory, the tissue engineering products with or without cells. What time frame we are talking about in terms of going, bringing it from clinical trials all the way to regulatory. How many years we are looking at?
Yeah. Well, okay. So for the first part of the question, so let me remember, what was the first one again? The in vitro, in vivo, yeah. So, so I think that is one of the big challenges. So I think in vitro studies are valuable, but they're not, they help you understand trends and what are important factors in my design. Is it degradation rate or is it that I need to have a factor present for a certain length of time. But I think you do have to go in vivo early and cycle back and forth. So I don't think it's a linear path that I figure everything out in vitro and then I go to small animal model and then I go to large animal model. But instead what we find is useful is that you do some in vitro experiments to help you narrow down formulations and then early go to the small animal model and you figure out, oh, I thought this was going to degrade in a certain way, but when I go in vivo, there's another enzyme present that really accelerates the breakdown of this, so I need to go back and change this part of my design. So that's what we find is really helpful. But it's still a long process to go through this. And so in our example with the cartilage, we started our work with cartilage in like 1998. And it went into humans in 2009. So that gives you an idea of the scale. And I think part of the reason for some of that difficulty, right, is just there aren't that many products. And at least with our Food and Drug Administration, everything's kind of classified as a biologic. So cells are biologics. The growth factors are all drugs. And then anything that's not a biologic or a drug is a device. And so materials are devices. So anytime you combine those, it's called a combination product, and they create a special path for that, and you have to work directly with them. But that's why I think maybe if we try to simplify and make it as simple as possible, and if we can start to think about systems that are just a material that could perhaps somehow recruit cells, or a material with a drug that's already used in that application, like rhBMP, so the drug's already approved, and I'm just trying to have a delivery device for it. I think as we start to have more of those, that it will help. And then I guess very finally, the last thing that I think I'll say is that I do think that in this field, there's also an unmet niche with all the advances right now in stem cells and iPS cells and all. But there's starting to come out some papers that show if I culture stem cells on tissue culture plastic, like many stem cell groups do and biologists, that when I transplant them, that they'll have a certain fate. But if I culture them in a hydrogel environment that's softer and more like the tissue, I can improve that fate. And I find that very amazing, that the context with which you ex vivo expanded cells and then transplanted them, the cells somehow it changes them in a way that even after you put them in a new in vivo environment, they can perform better. So I think there's also a lot that can be done with in vitro culture systems and helping in that manner too.
We also should have some questions from students.
Let me do one, and then they can think about what they're going to ask. So mine actually segues right into that, because I was going to ask some weird chemical stuff. So you're using self-hydrels in your light-activated link-up. Something that was extremely weird to me when I first heard it was that you can alter stem cell fate with beta-mercaptoethanol, or cause cells to go back to stem cell from more differentiated. Beta-mercaptoethanol didn't strike me as a likely compound, but maybe self-hydrels in a more general way are doing it. And so maybe part of the reason your matrix is doing stuff is because of weird signaling that we didn't even know was going on, right?
Is that the really recent?
No,
This is a while. That's long. Okay.
Long time ago.
Yeah.
And so there's some more stuff in between, of course. And then in terms of the culture, so with C. elegans embryonic primary cells, they're never passage, but they like peanut lectin. They don't like polylysine, but they like magic stuff that they put on them in Germany that they won't tell you what it is on the tissue culture, on tissue slides. So there's definitely a lot going on with what you put on the surface that the cells are adhering to, and I don't even know why they like peanut lectin. Or a variety of things in fetal bovine serum either.
Yeah. No, I find it a really interesting area.
I'm quite impressed by the video that you provide hydrogel to the cells, and first they can move, but then they move actively. And I do cell culture, and I know that I put some collagen on the cover slips, and the cell can spread, but they don't move. They don't move so obviously. So I am wondering if I provide hydrogel to cells, whether they will move. And do you know the determinant factor that regulates the move?
Yeah. Well,
to the hydrogel case.
Yeah. So that's a great question. So I think how cells migrate and move, whether it's on a surface or inside of a hydrogel, it actually depends some on the number, how strong the cell binds to the substrate as well. So you could envision it if there aren't many binders at all. It's kind of like the cell's trying to move on ice. And so I add more ligands, and so then it can begin to move faster because it can attach. But if I add too much, then it becomes like a sticky glue surface, and they can't move. So actually, many cell types, there will be sort of an adhesive ligand density that promotes higher motility. That makes sense. And then when you put cells in 3 dimensions, then many times they have to push through or degrade the matrix to be able to move as well as attach. So with collagen, if you're using low or high density, that can influence how fast a cell can move.
Thank you very much for your talk, and I have a small question because I'm not an expert in the hydrogel, so I want to know that because hydrogel absorb a lot of water, so will it swell when the environment has a lot of water? Because if you put hydrogel in your body, there's a lot of water, will it swell? Thank you.
Yes. So, so if you implant a hydrogel, so hydrogels will naturally equilibrate to have a certain amount of water. And so if I make my hydrogel so that the water in it is about the same as what it would equilibrate to, then once I make it, it won't swell more. But let's say if I made it with half of that equilibrium amount of water, and then I put that in the body, then it's going to swell to twice the amount of water and change its size.
So when you put it in the body, will you equivalent it or not? Because
Yeah.
you want it to be the shape just like the wound it will.
Yeah.
Or it
So-
will not contact well.
Usually we try to get to about the equilibrium water, but sometimes there are advantages to it being a little bit lower so that if the hydrogel swells a little bit more, it will really apply some force to the surrounding tissue so it won't come out or move. But, yeah.
And I recall there's a presentation I've heard that given by a doctor that he said that the hydrogel is very magic. When you put it in the wound of the bone, it will help the bone to regrow or something like that. He said that the pure hydrogel is good enough. If you put some growth factor, it's better, but it's just a little better.
Yeah.
I want to know that in your opinion, does some drugs or growth factors, will it have that magic effect, or just hydrogel is good enough?
Yeah.
Thank you.
So putting in the growth factor like the BMP can help, but I think what happens is that bone is sort of a special case as well, because our bones they're very vascular. And normally when you break a bone, bones can heal. And it's because it's so vasculature, and when it forms that kind of clot, that it immobilizes all these factors that are present in your bone marrow. And it tells resident cells to come in and heal. So I think what happens with a hydrogel is when you have a really big bone fracture and that clot kind of forms, the problem is that clot dissolves away too fast as the cells start to come in because it's so big, so it's not stable long enough. But if you put a hydrogel in place, what we think happens is that it helps stabilize that clot. That clot still forms, and there are all those growth factors that are naturally in bone marrow and other things that get immobilized in this hydrogel. So it's not that it's really just the hydrogel, but the hydrogel kind of takes advantage of sequestering all those factors that are naturally found in all the blood there.
Thank you for your talk. I have a detailed question about your permissive hydrogels for catalyst. So how's the procedure goes actually? So you took the cells from the patient and cast a hydrogel with or without the cells?
No, so the way we did our procedure was we had cells from the patient that were expanded, and then we loaded them up in a syringe that contained sort of a buffer solution that had our precursors for our hydrogel. And then we injected it, and we had to put a little membrane over the cartilage so we could contour the surface right, and then we exposed to light, and we had an absorber inside of our hydrogel form.
I see. So it actually casting during the operation?
Yeah, yeah.
Okay. Oh, but how about your experiments? Your experiments showing those optimizations, are they doing in vitro?
Yeah, so most of the results I showed you were in vitro, but then I did show a few that were in vivo in the goat's
Okay.
knee. Yeah.
But if you do in vitro optimization, then you need to provide those environments for the cell to grow, right, in the hydrogel. For example, medium growth factors.
Yes. Yeah. And so we use sort of a classic culture media, but this was a case that actually when we went into the joint and put it in the joint, almost in all the animal models we saw more robust tissue formation, because I think our culture, we don't get all the signals right.
Sure.
And when you put it in situ and grow it there, it grows better.
Okay, so my final question is that when you're doing those experiments, you're just soaking your gel in the medium. Am I correct?
Yeah.
Okay, yeah, I see. Thank you very much.
I think in the interest of time, we will have this one last question, and then we will conclude the official seminar, and then you can ask more questions. Okay, later. Okay.
It's a very simple question. I just wanted to ask if hydrogels can be used at the surface of the body or not.
On the surface of the body? Yeah.
on skin.
Yes, on skin. Yes.
And like if it's the case, can it make like the skin younger or something anti-aging?
Yeah, so it's being used or applied more for wound healing to try to prevent scarring or try to heal difficult wounds like diabetic ulcers. But yeah, unfortunately, I don't think that we figured out how to make old cells make young skin. Would be very popular.
I would like to have that. Okay, in the interest of time, I know there are many questions. Let's thank Professor Anseth, Kristi Anseth, one more time. First. Okay, so I know you have lots of questions, so I have anticipated this. So if you are faculty members, if you are interested, we have some time slots still left. You can email Zoe Chu, okay, at ust.hk. So she will try to schedule it into a time that is convenient for you, and Professor Anseth will be here until at UST campus until Wednesday morning. And for students, okay, we also have booked a room in room 4577. This Wednesday morning from 10 to 11 a.m. So you can have a time to chat with Professor Anseth about this research or other things, okay? Probably you have already very inspired by her work. So, so feel free to do that. Okay, thanks. Room 4577, that is lift 2728, is the conference room of the Department of Chemical and Biomolecular Engineering. Okay, if you have any question, you can also email Zoe or myself. Okay, I'm Professor Ying Chao. Thanks.